As you're joining in, we invite you to add in the chat where you're calling in from, where you're joining us from today, and you're welcome to add why you're here, what brought you to our webinar series. Okay, well, let's get started. Again, welcome to our Fermentation Friday webinar series. This webinar is coming to you from our EAT-LAC webinars. This is our third webinar in our series called The Science of Fermentation. I'm Maria Marco. I'm a professor in the Department of Food Science and Technology at UC Davis. EAT-LAC stands for Evaluating and Testing Lactofermentations Across the Country, and is the name of a project led by scientists in the UC Davis Department of Food Science and Technology. The goals of the project are to expand our knowledge of fermentation and provide accurate information related to production of fermented foods. We want to help you understand what you are buying and eating when you invest in fermented foods, and provide information that will encourage you to try fermenting at home. The webinar series is made possible by a grant from the California Department of Food and Agriculture's Specialty Crop Grants Program. We are using Zoom as the platform for this webinar series. We will be muting all participants and encourage you to ask questions in the chat function. Please keep your video off during the webinar, and we will address as many of the pre-submitted and live questions as possible at the conclusion of the presentation today. Following the conclusion of the webinar, we will send you a link for a survey in Qualtrics. Please take time to complete the survey and provide us with feedback. Today's webinar is the safety of fermented food. In this webinar, we will discuss how the fermentation process results in safe foods for human consumption. We will also discuss potential risks of a fermentation gone bad. We will begin with a 30-minute presentation from our speaker, followed by a short, approximately 5-minute video related to the producing fermented fruits and vegetables at home. After that, we will have approximately 15 minutes of questions and answers. It's my pleasure today to introduce our speaker, Dr. Aaron DiCaprio, a virologist and food safety specialist in the Department of Food Science and Technology at UC Davis and the UC Division of Agricultural and Natural Resources. Dr. DiCaprio's food safety extension program spans the farm to fork continuum, providing food safety information to growers, food processors, and consumers. She serves as the Food Safety Technical Advisor for the University of California Master Food Preserver Program and provides guidance at a local and national level on best practices for safe home food preservation techniques. So with that, I'll turn the mic over to Dr. DiCaprio.
Thank you, Professor Marco. Hi, everyone. Thanks for being here today for the last webinar in our series. So I'm going to be speaking about the safety of fermented foods. So let's go ahead and jump into it. So there are three basic types of hazards that can be present in food and lead to foodborne illness. The first is physical hazards. So these hazards include things like bones, glass, or wood that might end up in a food product and can cause damage to someone's teeth, their mouth, their digestive tract if they were to consume that product. The next type of hazards are chemical hazards. So these include allergens. They could include pesticide residues, processing aids, sanitizers that ended up unintentionally in a food product. When it comes to allergens, the big 8 allergens are dairy, eggs, peanuts, tree nuts, fish, crustaceans, wheat, and soy. There are certainly other allergens out there in the world, but these are the 8 allergens that food processors in the United States must include on their labels. And then the last class of hazards are biological. And so these biological hazards include bacteria, viruses, and parasites that can cause foodborne illnesses. So outlined here are the major biological, chemical, and physical hazards associated with home fermented products. So from the chemical hazard standpoint, we're concerned primarily about biogenic amines and allergens. The allergens, of course, come into play when you're using a starting ingredient that's allergenic. So if you're using milk, for example, that would be a concern. There is some evidence that fermentation can alter the proteins that cause allergic reactions, but this isn't very well established at this point in time. So it's important to take caution if you're fermenting foods that do contain allergens, if you suspect a person with allergies might consume that final product. Biogenic amines, as Professor Marco introduced last week, can be produced by some types of lactic acid bacteria. And if ingested, they can produce some inflammatory responses and headaches. In terms of physical hazards, the major thing I just want to point out here is that, remember, during the fermentation of many foods, carbon dioxide gas is going to be produced during that fermentation. And so you can have pressure build up in fermentation vessels if they are not vented and that and microbial growth is not controlled. So this can actually lead to shattering or exploding of fermentation vessels. There was actually just a recent recall of a beer product. It was a very fruit forward, sweet kind of beer where apparently they canned this beer and there was still residual sugar in the product. And so there was a secondary fermentation in the can and they had to recall the beer because the cans were exploding. So this is something to think about and take precautions against. And then what I'm going to focus on today are the biological hazards associated with fermented foods. And so the first biological hazard we're concerned about in fermented products is Clostridium botulinum. And then the second type of hazard is really a group of pathogens called enteric pathogens. So we'll spend time defining these microbial hazards more in depth today. So the focus of our project is fruit and vegetable fermentation. So that's going to be the primary focus of my talk today, but much of what I'm going to talk about in terms of the hazards and how we control for those hazards in fermented food is also going to hold true for other types of fermented products. But when we talk about fruit and vegetable ferments, there's really a very long history of microbial safety with traditionally fermented fruits and vegetables. However, there have been foodborne illness cases associated with small scale production of cabbage-based ferments, notably kimchi. So in 2011, there was a case of botulism that was traced back to some homemade kimchi. And in 2014, there were several outbreaks associated with Shiga toxin producing Escherichia coli or E. coli, traced to kimchi that was prepared and served in a school cafeteria in Korea. So I give these examples just to say, while outbreaks associated with fermented fruits and vegetables are rare, vegetable and fruit fermentation is not without risk. There can be issues related to biological hazards with these types of products, but these risks can really be mitigated by following recipes with proper salt levels, monitoring time and temperature during fermentation, using fresh ingredients that were produced in a manner to minimize microbial contamination risks and that are also fresh, and by following proper cleaning and sanitation procedures in the kitchen. So I want to briefly introduce the key organisms that are included in the field of food microbiology because it's relevant to what I'm going to talk about today. So the field includes diverse organisms from different domains of life. Parasites, fungi, and yeasts represent eukaryotic organisms that have a nucleus within their cells. These may be unicellular or multicellular organisms, depending on which specific type of microorganism we're talking about. Bacteria, in contrast, these are single-celled organisms that lack a nucleus within their cells. And viruses, which are kind of my area of specific expertise, are not actually considered living organisms as they're obligate intracellular parasites. So just to give you, a very high-level overview of the kind of microorganisms that are relevant to foods. So microorganisms are a very significant cause of food spoilage, and typically microbiological spoilage can be caused by bacteria, yeasts, and molds. There's also types of spoilage related to enzymatic activity or chemical alterations, but microorganisms are really the leading cause of spoilage of foods. Generally, spoilage microorganisms do not cause a food safety issue themselves. However, the growth of spoilage organisms in some types of foods might make conditions suitable for pathogens to grow. There are certainly also many beneficial microorganisms in the world. So obviously here we're very enthusiastic about fermented foods. So we know how important the microorganisms are to the production of these food types specifically. In addition to fermented foods, microorganisms are also used by the food industry to produce chemicals and enzymes that are then incorporated into the production of processed foods. So microorganisms not only important for fermented foods as a category, but they also play a really critical role in most of our processed food production. There are certain types of microorganisms that pose a health risk, and some of these are the causative agents of foodborne illness. So there's really multiple terms that are used to describe a sickness that arises from consuming a contaminated food or beverage, and I listed some of them here just for reference. There's really not one syndrome that encompasses all foodborne illnesses. I think typically we associate the symptoms of nausea, vomiting, and diarrhea with foodborne illness, but there are many other types of symptoms that can be associated with a foodborne illness beyond those three that we all typically think about. So this slide lists the types of microorganisms that are relevant to food, and they're categorized as either contributors to food spoilage, foodborne illness, or they're used for food enhancement through fermentation. So again, this is just very high level. We're talking about generally these groups of microorganisms. So from the food microbiology standpoint, bacteria really are the most important group of microorganisms because bacteria, certain types of bacteria are a food safety concern. There are many different types of bacteria that contribute to food spoilage. And of course, there are specific types of bacteria that are used beneficially for fermentation. Molds and yeast are also major contributors to food spoilage. So there are many kinds of yeast and molds that can lead to spoilage of food products. However, as already covered in this series, there are certainly yeast and molds that are used for fermentation. So there are also beneficial kinds of yeast and molds for the food industry. There are also specific types of molds that if they grow in certain food products can produce mycotoxins that if you were to ingest the foods that contain those toxins could lead to foodborne illness. This is typically limited to certain commodities like grains, corn, tree nuts, and peanuts. So, a mold that you see growing on a piece of bread in your kitchen isn't going to be something we're concerned about producing mycotoxins, but just to be aware that there are certain kinds of molds that if they grow in foods and produce these toxins, that can lead to foodborne illness. Viruses and parasites cause foodborne illness, but they don't contribute to spoilage or fermentation because they don't have the ability to multiply or grow in foods. Both viruses and parasites need a host like us to be able to increase in numbers, so they're not actually going to grow in food products. So for the microorganisms that do grow in food, bacteria, yeast, and molds, this graph represents generally the different states in which these microorganisms can exist in food. So on this graph, the horizontal axis is time in hours and the vertical axis is the number of microorganisms. So due to the fact that microorganisms can grow, meaning increase in number very rapidly, their count is represented on a logarithmic scale. So that means every change in unit on this scale is actually a 10-fold increase or decrease in number. So in this figure here, we kind of arbitrarily started our curve at 3 log or 1,000 CFU. So the unit CFU stands for colony forming unit, and it's typically per gram or per mil of food. So if you've ever seen a Petri plate, a picture of a Petri plate that's covered with little dots, those dots are bacterial colonies, and microbiologists will count those individual colonies and determine how many of these colony forming units are present per gram or per milliliter of food. So under ideal conditions, our bacteria, yeast, and molds can multiply very rapidly, and that's depicted by the blue line here labeled growth. So you can see within 8 hours, the number of, we'll just say bacteria in our food sample increased from 1,000 CFU to 100 million CFU per gram. So again, if conditions are ideal, we can see really rapid increase in numbers of bacteria as well as yeast and molds. When conditions are not suitable for growth, microorganisms may simply survive in a food product, so their numbers neither increasing or decreasing, and that's represented by the green line here. So we started with 1,000 colony forming units in our food sample, and that's where we were 16 hours later. It may also be possible that conditions in a food are harsh or unfavorable. And in this case, we can actually have microorganisms dying or being inactivated in food, and that's depicted by the red line here. And so when we're talking about fermented foods, we've already spoken a bit about how the growth of fermentation microorganisms, one of the byproducts is production of acid or CO2, which has the side effect of lowering the pH or increasing the acidity level of a food product. And so we know that, high acid levels do have a negative impact on the survival and growth of some of the pathogens of concern. And so, in a fermented food product, what we want to see for foodborne pathogens or spoilage microorganisms is have them die off due to high acid levels. In some cases, we'll talk about specifically one pathogen where we simply want to inhibit growth of that pathogen in a fermented food through, again, decreasing the pH of that product. So more on that to come. So I want to define a little bit now the potential biological and, again, essentially just microbial hazards that can occur in fermented foods. So the first is Clostridium botulinum. So this particular bacterial species can produce a neurotoxin if it grows in food. And if someone or an animal would consume that food product that contains the toxin, it leads to the disease botulism. So Clostridium botulinum only grows under oxygen-free conditions and in low acid foods. So botulism can potentially occur if a fermentation is not successful and the acid levels remain low in that fermented product. Enteric pathogens are a group of foodborne pathogens that include bacteria, such as Shiga toxin-producing Escherichia coli, Salmonella, and also includes viruses and parasites. So these enteric organisms can end up causing gastrointestinal disease. Oh, excuse me. So these organisms can end up causing disease if proper sanitation practices are not followed, and if the fermentation for one reason or another is not successful. So what's happening during lactofermentation to make food safe? So Professor Marco covered this already, but as lactic acid bacteria grow, the bacteria convert sugars into lactic acid, acetic acid, and CO2. During growth, the bacteria also produce antimicrobial compounds, and that keeps bacteria competing for their nutrients at bay. These antimicrobial compounds can also help to control for the growth of microorganisms that might spoil a ferment, and they can potentially inactivate some pathogens if they're present. What's really critical for ensuring the safety of a fermented food is the production of organic acids and carbon dioxide, which have a pH lowering effect on the food. So lactic acid and acetic acid are both organic acids. This lowering of the pH can inhibit the growth of some pathogens and directly inactivate others. These organic acids themselves are actually frequently used as food preservatives by the food industry because they have very strong antimicrobial effects. Another side effect of lactic acid bacterial growth is limiting spoilage and potentially impacting safety is the fact that these lactic acid bacteria, they're going to be utilizing sugars present naturally in the fruits and vegetables for their own metabolism. And this makes those carbohydrates unavailable for other microbes to use for their own metabolism. So in this way, the lactic acid bacteria outcompete other microbes present in a fermentation for nutrition sources and inhibit their growth. Since pH lowering is essential for the safety of fermented foods, I just want to refresh everyone's memory on pH. So pH is the negative log of hydrogen ions in a solution, and it's essentially the measurement we use to determine acidity levels. So pH is measured on a scale from 0 to 14, and this is on a logarithmic scale. So again, each unit change represents a 10-fold change in the hydrogen ion concentration. A pH of 7 is considered neutral. pHs below 7 are acidic, and pHs above 7 are basic or alkaline. So I just wanted to give some examples of pHs of different foods. So in this example, the pH of a black olive is 7, where the pH of our lemon is 2. And in practical terms, what this means is the lemon is 100,000 times more acidic compared to the black olive. So that's the difference in that hydrogen ion concentration between something that has a pH 7 and something that has a pH of 2. So when we talk about the pH of foods, the pH of most foods is actually below 7. There are only a few types of foods that have a true neutral or basic pH. So some examples include black olives and hominy, because these products are actually lye cured as part of their processing. So lye is very basic, and exposing what will become hominy and the olives to that lye increases their pH significantly. Egg whites also have a slightly basic pH. However, pretty much everything else has a pH below 7. So when we talk about high acid and low acid foods, we have a different dividing line for what distinguishes a high acid and low acid food, and that's a pH of 4.6. And this is because at a pH of 4.6 or below, the spores of Clostridium botulinum are not able to germinate and produce toxin. At pHs above 4.6, Clostridium botulinum spores can germinate and grow. So just generally, fruits are going to be naturally high in acid, while vegetables, meats, and dairy, those are going to be considered low acid foods. So I just mentioned Clostridium botulinum spores, so I want to take a few minutes just to describe vegetative bacterial cells and bacterial spores. So an actively dividing and metabolizing bacterial cell is said to be in what is called the vegetative state, and this is essentially the active growing state. So all bacteria can exist in the vegetative state. Some types of bacteria, not all, have the ability to form what's called a bacterial spore. And the spore forms when conditions become harsh, and a vegetative bacterial cell would not be able to survive. So this bacterial spore is very hardy in the environment and can survive very hot, dry, and acidic conditions. But this spore is essentially kind of an inert particle out in the environment. It doesn't have active metabolism. It's not dividing, but it is able to sense if environmental conditions improve to a level that would allow for vegetative cells to survive and divide. And in this case, that bacterial spore is able to germinate and again enter the vegetative state. So again, some bacterial species have the ability to form spores, but not all. So Clostridium botulinum is a spore-forming bacteria that's found naturally in soil. So we just assume all foods potentially harbors this particular bacteria since it's found naturally in soil. This bacteria is a strict anaerobe, meaning oxygen is toxic to the bacterial cell. And so Clostridium botulinum will not grow on most foods due to oxygen exposure. So if you pick up an apple, you don't have to worry about there being Clostridium botulinum toxin on that apple because it's exposed to oxygen. Clostridium botulinum is not able to grow also when acid levels are high. So remember again, pH of 4.6 or below is going to inhibit the growth of this bacteria. So during the fermentation process, we aim to create a low or no oxygen condition so our lactic acid bacteria can grow well. However, this provides an anaerobic environment where Clostridium botulinum can potentially grow if sufficient acid is not produced during fermentation. So Clostridium botulinum produces a potent neurotoxin as it grows called botulinum toxin. And if that toxin is ingested, the resulting disease is called botulism. The typical symptoms are listed here, and they can appear anywhere from 12 to 36 hours after ingestion of the contaminated food. This disease can be fatal within days if antitoxin is not administered. And it's important to note that even those that receive the antitoxin are often hospitalized for weeks or months due to the effects of being exposed to this particular toxin. The other types of pathogens that we're concerned about in fermented foods are enteric organisms. So these microorganisms are typically found in the gastrointestinal tract of humans and animals. As such, we historically associated them with products of animal origin, such as beef, poultry, pork, and eggs. However, many other types of foods can easily be cross-contaminated with enteric pathogens. So we've seen, numerous outbreaks in produce due to contamination with enteric pathogens. Important to know, these pathogens are not spore formers, so they don't have the ability to form those really environmentally hardy spores. But many of these pathogens are able to survive and grow at pHs below 4.6. So that 4.6 is kind of the magic number for Clostridium botulinum, but many other types of foodborne pathogens can survive and even grow at pHs below 4.6. So here's a list of some enteric pathogens of concern. So again, I'm kind of honing in on fruit and vegetable fermentations here. So there have been outbreaks associated with fruits and vegetables used commonly in lacto fermentations caused by bacteria, parasites, and viruses listed here. However, the outbreaks with bacteria have really far outnumbered the outbreaks we've seen in produce caused by parasites and viruses. Also, these bacteria, they do have the ability to grow in food, unlike viruses and parasites. So these bacteria are really kind of our primary hazard of concern for fermented fruits and vegetables. I will also kind of inject here, all of the pathogens on this list could be introduced to a ferment if proper cleaning and sanitation practices aren't followed. So, someone that has symptoms of gastroenteritis or is otherwise ill shouldn't be preparing foods. If you handled raw chicken in your kitchen on a counter and then you didn't sanitize that counter and then set down some equipment you were going to use for fermentation, you could easily have cross-contamination with some of these enteric pathogens. But of the bacteria listed here, Shiga toxin producing E. coli have been determined to be the most acid tolerant in fermented and acidified vegetables. And so that's really, commercially, when someone's developing a a validated fermentation process, they're typically going to be looking to see that sufficient acid is produced during fermentation to inactivate some of these acid tolerant STECs. Our traditional vegetable fermentations, things like fermented cucumbers, sauerkraut, kimchi, they've all been shown to produce sufficient acid to inactivate the Shiga toxin producing E. coli. So from a safety standpoint, sufficient acid production is the critical control point for ensuring the safety of a fermented fruit or vegetable. So our fruits and vegetables, they have natural sugars present that are going to be exploited for our fermentation purposes. So cucumbers, they typically have around 2% sugar by weight. Cabbage can have 4% or greater. So these traditionally fermented vegetables, they do have sufficient sugar for the lactic acid bacteria to grow and lower the pH to well below 4.6. And typically when the fermentation is finished, the pH is going to be around 3.2. And at the end of fermentation, when we have that very low pH, the lactic acid bacteria have actually utilized almost all the sugar in those traditional vegetable ferments. So there's almost no sugar remaining, which also helps inhibit the growth of any spoilage microorganisms. So acid production during fermentation is key. So how can we ensure that sufficient acid is produced? So this is just kind of a little cartoon visual of what is happening microbially in our lacto-fermented fruits and vegetables. So in our raw ingredients, we're going to have a really diverse microflora. So we'll have high levels of potential spoilage microbes, low levels of lactic acid bacteria. This could be as low as 1% of the population, and potentially low levels of pathogens. During fermentation, the lactic acid bacteria are going to start metabolizing sugars. They'll increase in number and they'll be producing acid as a byproduct of that metabolism. So this is going to help inhibit the proliferation of spoilage microorganisms as well as our pathogens. And then eventually the lactic acid bacteria are going to dominate the fermentation, and the conditions of high acid will inactivate or inhibit. They'll inactivate enteric pathogens or inhibit Clostridium botulinum spores from germinating. So this level of acid coupled with the lack of nutrient sources is also going to inhibit many spoilage microorganisms. So one key component to ensure this progression to a lactic acid bacteria dominated fermentation is to add salt. So the appropriate level of salt needed is going to vary depending on your starting ingredient, but it can range anywhere from 1 to 15%. This variation is based on the level of sugar present in the raw ingredients, the lactic acid bacterial species responsible primarily for the fermentation, and also on whether or not there are any specific enzymes we want to inhibit in the vegetable during fermentation. Sauerkraut fermentation, for example, salt concentrations are typically 2.25 to 2.5 in commercial sauerkrauts. For cucumber pickles, low salt pickles, salt concentrations range from 3 to 5%, and high salt pickles that can be anywhere from 5 to 16%. So these levels of salt don't necessarily inactivate or inhibit other organisms, but the lactic acid bacteria grow better under these high salt conditions compared to other microorganisms. So the addition of salt gives the lactic acid bacteria kind of a leg up to outcompete other microorganisms early during fermentation. And then once they begin to metabolize sugars and produce acid, this further establishes a foothold for the lactic acid bacteria in the fermentation. So lowering salt concentrations can reduce the likelihood that lactic acid bacteria will be able to dominate the fermentation, and this can increase spoilage. And in some cases, this can result in insufficient acid production, which can lead to potential food safety issues. So time and temperature is really important to ensuring sufficient acid is produced. And also for our enteric pathogens, the time those pathogens are exposed to acid is really important for their inactivation. So in a home fermentation where we're simply using a saltwater brine, the starting pH of that brine is going to be around pH of 6. But a properly prepared and managed vegetable fermentation will typically have a pH of less than 4.6 within a week after the start of fermentation, but it might take a little longer depending on the vegetable matrix and the temperature at which we're fermenting. This pH will continue to drop below 4.6. And in cabbage-based ferments, it can take up to 3 weeks to have sufficient acid production and pH lowering to inactivate enteric pathogens of concern. So for commercial fermented fruit and vegetable processors, they have to closely monitor pH reductions during production, ensuring that that pH drops below 4.6 within the first few days after a fermentation is started. And they have to continue to monitor that pH even after it drops below 4.6 to ensure the pH keeps going lower and there's not any unexpected increases in pH due to the growth of spoilage microbes. For those fermenting at home, it's important to follow a recipe that specifies a specific time and temperature range for fermentation to be complete. Short fermentations of a week or less for fruit and vegetables. These are going to likely be considered lightly fermented products. And so sufficient acid might not be produced in a short period of time to levels to be able to control for enteric pathogens. And I'll just also interject here, there are some types of lactic acid bacteria that aren't going to be producing CO2 gas actively as a byproduct. And so I've seen a lot of recipes where they say you can, your fermentation's done when there's no more active bubbling, but, there may be cases where you have continued pH lowering even after you've stopped seeing those active signs of bubbling in a ferment. So important just to, if you can, start with a research-based recipe that's specifying a specific temperature range and length of time for that fermentation to ensure the safety of the product. So the critical factors really to consider to ensure a safe fruit and vegetable fermentation are first, using high quality raw ingredients. So these should be ingredients that haven't begun to spoil. If there are high numbers of spoilage microorganisms, to start with, it'll be really difficult for the lactic acid bacteria to dominate the fermentation. It's also really important that any raw ingredients are produced and harvested in a way that minimizes risks of cross-contamination with enteric pathogens. It's also important that you take care when preparing ingredients. There are certain parts of ingredients that might need to be removed because they can contain enzymes that may negatively impact a fermentation. But it's also really important in preparation to have food particles that are uniform in size. Typically, Smaller slices or shredded food particles will release more water and nutrients, and that promotes the growth of lactic acid bacteria. Salt concentration, really critical. We've just talked about how important adding salt is for promoting lactic acid bacterial growth in these ferments. Important to ferment at an appropriate temperature. So typically those temperatures are going to be between 65 and 75 degrees Fahrenheit. So going much below those temperatures, you might not get fermentation. If you go above those temperatures, you increase the likelihood that you'll have growth of spoilage microorganisms. And then allow adequate time for the fermentation to complete. So it takes a while for significant acid to be produced. So be patient, and again, follow the directions in the recipe. For most of our fruit and vegetable ferments, it's going to be anywhere from 3 to 6 weeks for the fermentation to complete. So I just wanted to point you to a couple references beyond eatlac.org. So I help manage the UC Food Safety website with Dr. Linda Harris, and we have a whole page dedicated to home food preservation. So there are some resources there related to fermentation, but generally, if you're interested in food preservation, we link to research-based recipes there. We also have a series of short YouTube videos related to safely preserving at home. So if you're interested in the wide field of food preservation, so beyond just fermentation, I recommend checking those out. And then listed here are just some of the scientific literature references I use when putting together today's presentation. I want to say a big thank you to our EatLac project team. It's been a hard year for everyone, but it's been really great to work with this team and see the energy they have for the project and the enthusiasm they have for fermented foods. So I just want to say thank you to the group for all their support over the past year. And so with that, I do have a short video I wanted to show. So I'll go ahead and do that now. And then with any remaining time, we'll address questions. So I did see the chat lighting up quite a bit. So I think we have some good discussions. Let me... Understanding the difference between a successful ferment and a spoiled one can be a challenge the first few times you ferment at home. To help ensure safety and quality, we recommend using fruit and vegetable fermentation recipes available from the USDA Complete Guide to Home Canning, National Center for Home Food Preservation, or extension publications from land-grant universities. Salt concentration is very important in fermentation. Therefore, salt amount should be measured carefully and a tested recipe should be followed. Salt concentrations in vegetable and fruit fermentations can vary from 1 to 15% and are specific for the vegetable or fruit being fermented. In general, lower salt percentages will favor the growth of more types of fermenting microbes. But lower salt concentrations can allow for mold and other contaminants to grow, resulting in a softer texture or an off-flavored final product. Salt concentration is also important in fermentation because it helps with water and nutrient release from plant tissues. Salt also can inactivate enzymes that can lead to the development of off flavors or soft textures. For example, salt is very important for inhibiting the activity of the enzyme pectinase. This enzyme degrades pectin, which is one of the structural components of plant cell walls. By inhibiting the activity of pectinase,
Salt helps inhibit softening of fruit and vegetable ferments. Beyond that, salt is also important for flavor development. It is important to take care in preparing raw ingredients and follow recipe instructions. Some ingredient components can negatively impact fermentation. For example, including the core of a cabbage can lead to the production of a sauerkraut that has a slimy texture. This is because the inner core of the cabbage has higher concentrations of sucrose compared to the cabbage leaves. The high level of sucrose can promote the growth of a type of lactic acid bacteria that produces dextrins as they grow, and these dextrins can make the sauerkraut slimy. Another example is softening of pickles due to enzymes found in the blossom end of a cucumber. The take home is that many of the directions found in fermentation resources are critical to the success of the fermentation, so follow them. There are some obvious signs if a ferment has spoiled. A fermented fruit and vegetable should not smell putrid, so if a ferment has a strong odor that is not characteristic of the final product, it should be discarded. Sometimes there are obvious color changes that indicate spoilage. For example, if you start with green cabbage to make sauerkraut and it turns pink, that is a sign of spoilage. Generally, anytime you see a dark colored growth in a ferment, that is a bad sign. If you see dark colored molds at the brine and air interface, it is time to discard the ferment. Also, if you notice extensive mold growth throughout the contents of the fermentation vessel, it should be discarded. Some types of spoilage microbes can consume acid as they grow, and this leads to potentially unsafe conditions where foodborne pathogens may be able to proliferate. There are some types of visible microbial growth that you may see on a fruit or vegetable ferment that are common. For example, in sauerkraut, it is common for a white or gray film to form on the surface of the sauerkraut at the brine and air interface. This is typically yeast that grow in lower acid conditions but need air in order to increase in numbers. You can safely remove this white film and continue to ferment. However, you want to monitor this closely and remove the film every few days so it does not build up to high levels. Yeast is also important for the fermentation of olives. You may see yeast bubbling on the surface of a brine used to ferment olives. It will be white in color. As with the sauerkraut, it is important to monitor the surface of the brine for yeast and remove it periodically. Anytime you are using a tool to remove surface yeast, make sure your utensil is sanitized to minimize risks of contaminating your ferment with other unwanted microbes. Unfortunately, there is not a great way to test and see if a home fermentation is safe. You can feel confident about the safety of a ferment when you follow a research-based recipe and see signs that fermentation is progressing. If you notice signs of spoilage, the ferment should be discarded because there might be a food safety risk in addition to the fermented food not being tasty. You can find fermentation recipes and facts on the EAT-LAC website or by following the project on social media. Understanding the difference between a success.
All right. I think I talked too long. I get very excited about food safety, but maybe we have time for one or two quick questions before we wrap up.
Thanks so much, Erin. That was really wonderful. And indeed, we did receive a large number of excellent questions in our chat. And we know we're not going to be able to do them all justice here in what time we have remaining. But I wanted to come back to a few points. Actually, right from your video in your last few slides, you mentioned that we don't have great ways to know whether our fermented fruits or vegetables are safe. Are there certain measurements or indicators we can use at home to assess this?
Yeah, it's a really great question. And I think there were a lot of questions related to that were pre-submitted and that came in the chat as well today. And so, from the commercial side, they have a lot more options when it comes to testing, right? They probably have pH meters where they're monitoring pH in real time of the fermentation and can see that trend. My pH is going down. They may also be doing their own microbial tests, right? Where they're pulling out samples periodically, and whether they're testing in their own lab or sending that out for analysis. So on the consumer side, we try to advise that, the best approach is to start with a recipe that has some research behind it, that we feel confident that if we follow these kinds of processes, that we're going to have a product that has a safe final NPH. There are ways that a consumer could look at pH at home. So there's, pH test strips that are relatively cheap, easy to use. Those aren't the most accurate. It gives you a general sense of the pH range. There are also pH meters available that won't break the bank. They're not exorbitantly, expensive. But I will just kind of say that, pH meters aren't necessarily using them as an intuitive. So they have to be calibrated. You have to take care of that pH meter and make sure it's electrodes stored in a way where your measurements are going to be accurate. So, generally, I think for a consumer starting out, visit eatlac.org. We have a couple of recipes up there for sauerkraut and fermented cucumbers that, were developed by food safety specialists. And so that's a good place to start and feel confident that you're going to have a safe end product. So yeah, there's no good way to look at a ferment though, and other than if there's growth, mold or yeast growth there, that it's good or bad.
Yeah, we had also quite a few questions on how to recognize if the fermented food has spoiled. And you touched on that a little, but maybe you want to repeat or elaborate on it, not just for safety, but spoilage.
Yeah, and I know you have a lot of experience with the industry too, so you probably have some things to add. But generally, I think it can be obvious when things have gone wrong, right? So that picture of the olives I showed where it had that kind of blue-green mold all over the surface, I think pretty much everyone would say it's time for this to go. I think it's very common to see, that white gray film on the surface, some, other evidence of yeast growth. And, if it's not everywhere, it's completely reasonable to scrape that off periodically. You want to check your ferments every day if you can. Don't let them go for 4 weeks and then take a peek in to see if there's yeast. You've got to maintain them. And so, if it's a small amount of that white gray color yeast, go ahead and remove that. Typically we say if you see anything that's dark in color, that's a bad sign. So if you see anything that's black or, even in blue, that's probably not something normal. And it's time to consider, stopping that particular ferment. There also shouldn't, fermentation has a distinctive odor, right? It's not something that smells, putrid, rotten, really. It's so I think smell is another good indicator if there's anything that is a smell you wouldn't typically associate, with a sauerkraut, for example. That's another sign that you have a spoilage issue on your hands.
Yeah, to use our senses. Probably evolved to recognize bad food and to avoid it, or through these experiences. So I think we're out of time, Erin. But I want to, for everybody here, to point you to the link that Erin shared in the chat. It's really important that we receive feedback on these webinars, and as we plan what we'll be doing next in our work. And yeah, so please do go to that link. We hope the information presented in today's webinar and throughout our webinar series was informative. Please take the survey now if the time permits. We will also send an email with the survey later today. The short video that was shown and a full recording of today's webinar will be made available on the EAT-LAC project website, eatlac.org, and the UC Food Safety website, UC Food Safety at ucdavis.edu, and this will be in the next few days. And Erin, I'll turn over the remainder to you.
Yeah, so I want to thank you all for joining us for the webinars and for your enthusiasm about the science of fermented foods. It seems like we have a lot of experience in the group just from the kinds of questions that were asked, so we really appreciate it. So if you are someone that's making your own home fermented fruits and vegetables, or if you have a business where you're making fermented fruits and vegetables, we'd love to have a sample from you for our citizen science project. So the instructions for getting a sample to us are at eatlac.org. And I also want to say, along with developing some new research-based processes for home fermentation and evaluating home ferments through the Citizen Science Project, we're also about to launch a survey related to fermented foods. And it's aimed to understand current fermented food consumption habits and patterns, and determine how many people are fermenting at home and if any of their home fermentation practices have changed at all due to the pandemic. So I know many of you said you'd like to get updates from us via email. So we do have a newsletter that goes out via email. We don't send it every week or anything like that. But our next newsletter that we send out will include this particular survey. So we would really appreciate you participating in that, letting us know what kinds of fermented foods you're eating now, why you eat those foods and what you're making currently. So keep an eye out for that. We'd really appreciate your participation in that research as well. So again, thanks for attending. The webinars will be posted online so you can view them again in the future if you like. And I just want to say thanks and have a great weekend, everyone.
Thank you.